There is a sentence in the standard medical literature on Toxoplasma gondii that has shaped how doctors talk to patients, how funding agencies allocate money, and how an entire field thinks about a parasite living inside roughly 2 billion human brains. The sentence goes something like this: latent infection in immunocompetent individuals is considered asymptomatic, harmless, no clinical significance. That sentence has been in the textbooks for decades, and since 1992, a single scientist in Prague has been quietly producing data that suggests it is wrong. Start with the parasite itself, because the biology matters. Toxoplasma gondii is a single-celled organism first described in 1908. It can infect virtually any warm-blooded animal, but it can only sexually reproduce in one place, the intestinal lining of cats. This is important. The entire life cycle of Toxoplasma is oriented toward getting inside a cat. Cats shed the parasite's oocysts in their feces. Oocysts get into soil, water, vegetation. Other animals, rodents, livestock, birds, humans, ingest the oocysts and become infected. After a brief acute phase that feels like a mild flu, the parasite goes latent. It forms cysts, tiny dormant capsules, primarily in the brain and in muscle tissue. These cysts persist for the lifetime of the host. No symptoms, no fever, no obvious sign that anything has changed. Depending on the region, somewhere between 30 and 70% of the population carries these cysts right now. The parasite needs to get from an intermediate host, say a rat, back into a cat. The most efficient way, the cat eats the rat. In a normal rat, this is unlikely. Rats have an innate, hardwired aversion to the smell of cat urine. Not learned behavior, genetic, deep, non-negotiable. A rat that smells cat urine runs every time, unless the rat is infected with Toxoplasma gondii. In 2000, a parasitologist named Joanne Webster published a study titled, without much subtlety, Fatal Attraction. Webster tested infected and uninfected rats in an enclosure with 4 scent zones: water, rabbit urine, the rat's own bedding, and cat urine. Uninfected rats avoided the cat urine zone entirely. Infected rats did not just lose their aversion, they actively spent more time there. The fear was not reduced, it was inverted. Later studies went further. In 2011, researchers showed that the specific brain pathway activated in infected rats during exposure to cat urine was not the fear circuit. It was the sexual arousal circuit. The parasite was not just dampening fear, it was hijacking the wiring that controls desire. This is not a metaphor. This is measured neural activity in a laboratory. The mechanism, the how, centers on dopamine. The Toxoplasma genome contains a gene for tyrosine hydroxylase, the enzyme that controls the rate-limiting step in dopamine synthesis. The parasite carries its own instructions for manufacturing the chemical that regulates reward, motivation, and risk assessment in the mammalian brain. Infecting dopaminergic cells with Toxoplasma enhances dopamine release several-fold. In late 2025, a study in Nature Communications confirmed the link experimentally. Engineered parasites with elevated enzyme expression caused stronger behavioral changes in rats in a dose-dependent manner. The more enzyme the parasite produced, the more the rat moved toward cat urine. In rodents, the science is clear, but rodents are not the controversial part. The controversial part begins in Prague in 1992, in a basement office at Charles University. Jaroslav Flegr was an evolutionary biologist in the parasitology department, a quiet, meticulous researcher who had contracted Toxoplasma himself during a year spent studying in Tokyo. The infection was latent, textbook harmless. But Flegr, who had studied the manipulation hypothesis in rodents, could not stop asking, if Toxoplasma rewires a rat's brain to serve the parasite's needs, what, if anything, is it doing inside 2 billion human brains? The standard answer was nothing. Humans are a dead-end host. The parasite cannot complete its life cycle by manipulating a human into being eaten by a cat. Therefore, any behavioral effects would be accidental spillover, evolutionary noise. The logic was clean, simple, and nearly universally accepted. Flegr disagreed, not because he had a competing theory, but because he thought the question had never been properly tested. His first studies were modest. No large grants, no dedicated lab team. He tested university staff and students for Toxoplasma antibodies and mapped their personality profiles using a standard psychological questionnaire. In 1994, he published his first paper. The results showed consistent differences between infected and uninfected subjects. Infected men scored lower on willingness to follow group moral standards, more suspicious, more guilt-prone. Infected women showed a different pattern, higher warmth, higher trust. The differences were not enormous, but they were consistent. And in a separate group of men diagnosed with toxoplasmosis over the previous 13 years, the magnitude of the personality shift correlated with how long someone had been infected. The longer the infection, the more the personality had drifted. A dose-response relationship, the signature of causation, not coincidence. The field dismissed it. Personality questionnaires are blunt instruments. Self-reported data is unreliable. The sample sizes were small. The premise that a parasite could meaningfully alter human personality struck most researchers as somewhere between unlikely and absurd. These were not unreasonable criticisms. Flegr acknowledged most of them in his own papers. He kept publishing anyway. A second study with a larger sample confirmed the pattern. Then Flegr moved from personality to reaction times. Infected subjects showed measurably slower psychomotor performance. Then came the traffic accident study, and the data stopped being easy to dismiss. Flegr compared infection rates in people who had been involved in traffic accidents with residents from the same area. Infected subjects were nearly 3 times more likely to have caused an accident. At the highest antibody levels, the risk was 16 times higher. The study was replicated in Turkey. A later meta-analysis across all available data confirmed the direction. Smaller effect size, but statistically significant, pointing the same way. During this period, Flegr was largely working alone. Funding was scarce. The topic was considered fringe at best, embarrassing at worst. He sat in his basement office, walls lined with bookshelves stacked perilously high, and ran studies on whoever would volunteer. Tenure committees reward safe choices. Grant committees fund established questions. As some colleagues quietly suggested, perhaps the researcher's obsessive focus on a brain parasite was itself a symptom of the brain parasite. Then an American psychiatrist named E. Fuller Torrey called. Torrey was one of the most prominent schizophrenia researchers in the United States, and he had been independently investigating a connection between toxoplasma and mental illness. Schizophrenia rates were higher in regions with higher toxoplasma prevalence. Mothers of schizophrenic patients had higher rates of antibodies, and dopamine, the neurotransmitter most associated with schizophrenia, was looking more and more like the mechanism through which toxoplasma altered behavior. Torrey recognized Flegr's work for what it was: careful data, ignored by an establishment that found the implications uncomfortable. He said of Flegr, "If you want to become a full professor, you avoid controversial subjects." In science, that sentence explains more than most policy papers. The same pattern that Flegr documented in humans, recklessness, reduced fear, altered risk assessment, kept surfacing in other species, other contexts. Off the California coast, toxoplasma-infected sea otters were nearly 4 times more likely to be attacked by sharks than uninfected otters. Cats do not eat sea otters. Sharks do not carry toxoplasma. But the behavioral fingerprint was there. Infected animals behaving as if the cost of danger had been quietly recalculated downward. In 2018, researchers found that college students who tested positive for toxoplasma were more likely to major in business. Among professionals, seropositive individuals were nearly twice as likely to have started their own companies. Entrepreneurship correlated with a brain parasite. The researchers were careful about causation, but the accumulation of correlations across species after species, data set after data set, was becoming difficult to wave away. The popular version of toxoplasma is a cat parasite that causes mild flu and is otherwise harmless. The record says something more complicated. Not that toxoplasma is a puppet master pulling human strings. The data is not that clean. The effect sizes in humans are modest. Confounding variables remain. But the weight of evidence is no longer compatible with the one-sentence dismissal that has sat in textbooks for decades. A parasite classified as benign is producing measurable, replicable behavioral effects in multiple species, including humans. The problem was never that the data was hidden. Flegr published in peer-reviewed journals. The traffic accident data was replicated in other countries. The schizophrenia connection drew on over 20 independent studies. None of this was secret. The problem was that the data contradicted a comfortable consensus, and comfortable consensus in science has an immune system of its own. Funding committees do not reject uncomfortable findings explicitly. They simply fund something else. Tenure committees do not penalize controversial research directly. They reward safer choices. The result is a slow, structural drift away from questions that make people uneasy. This is not conspiracy. It is something more ordinary and harder to fix. The quiet inertia of institutions that reward conformity over curiosity. The same pattern, buried not by secrecy but by indifference, that runs through every story this channel has told. Flegr is toxoplasma positive himself. He mentions this calmly, without drama, sitting in his basement office surrounded by bookshelves stacked high enough to be hazardous. He has spent more than 30 years on this research. In early 2025, he published a 30-year retrospective, tracing the path from the first personality questionnaires to the molecular biology of dopamine manipulation. The question he cannot answer, the question nobody can answer yet, is the one that matters most. If toxoplasma alters human behavior, how much does it alter? A little? Enough to notice in a laboratory but not in a life? Or enough to shift the statistical distribution of personality traits, risk-taking, reaction times, and mental health outcomes across 2 billion people? The difference between those two answers is the difference between a footnote and a public health emergency. The honest answer, as of right now, the data keeps accumulating, and the textbook sentence, asymptomatic, harmless, no clinical significance, keeps looking less like a conclusion and more like an assumption that nobody bothered to test. Flegr was asked once whether the parasite might be influencing his own behavior, whether his obsessive, decades-long focus on a single organism against institutional resistance might itself be a symptom. He considered the question seriously and said he could not rule it out. Not because it's a joke, because it might not be. Next time on Buried Record, a different kind of obsession. A Japanese zoologist named Tsunemi Kubadera spent decades searching for an animal that had never been photographed alive. The giant squid. 200 years of myth, 0 confirmed sightings. The cost of that search was more than funding. That story is coming.